So welcome everybody. It's good to see you all again at the UW Astrobiology Colloquium. I'm Mike Wong, a postdoc in astronomy and astrobiology, and I'd like to begin as always with our land acknowledgement. We acknowledge that the UW Astrobiology program resides upon the land of the Coast Salish peoples, land that touches the shared waters of all tribes and bands within the Duwamish, Puyallup, Suquamish, Tulalip, and Muckleshoot nations. We recognize that we, the UW astrobiology community, benefit from the occupation and use of this land, and that it is our responsibility to mend and forge our relationship with Indigenous peoples who are extant and vibrant members of the broader Seattle community. And I'll put some helpful links to resources in the chat if you'd like to learn more. Okay. So it is now my pleasure to introduce you all to Professor Tanja Bosak. Dr. Bosak is from Croatia originally, and she earned her PhD from Caltech in the burgeoning field of geobiology, studying the signatures of microbial processes in ancient sedimentary rocks. She then spent 2 years at Harvard as a microbial initiative postdoctoral fellow, and in 2007 joined the Department of Earth, Atmospheric, and Planetary Sciences at MIT, where she is now a professor of geobiology. Among her large list of accolades include the Subaru Outstanding Woman in Science Award from the Geological Society of America, the James B. McElwain Medal from the American Geophysical Union, and the Harold E. Edgerton Award for Outstanding Achievement in Research, Teaching, and Service to the MIT community. Now, I also heard that she recently testified to Congress about the Perseverance rover as the Mars 2020 mission's returned sample science co-lead. So we're very delighted to have her. I think this means that we are just as important as Congress. Maybe you already thought that, but anyway, that's my takeaway from all of this. So let's turn it over to Tanja now to tell us all about cyanobacterial evolution. Tanja, the floor is yours.
All right, you seem to have spies everywhere. All right, let's share the screen. Yeah, thanks so much for inviting me. I had some really nice conversations already with the astrobiology program, and clearly there are so many people who care about oxygen as my favorite molecule, and they care about the detection of oxygen on other planets, around other suns, but are really aware of the importance of this molecule and the process, biological process that makes it on our planet, which is photosynthesis. And so today I will tell you about our attempts to understand the molecular and fossil records of cyanobacteria, the first organisms that invented this process, oxygenic photosynthesis, when that happened and what it did for the evolution of the environment later on. So I'll be talking partly about the molecular record of cyanobacteria and what we can, how we can use molecular clocks to understand the evolution when all of these things happened. And then talk about a fossil record and how cyanobacterial fossils can tell us something about environmental conditions once cyanobacteria actually evolved. So before I go into that, there is a large team of people who have contributed to this work, and here is just many photos, so you can put some faces to names that I'll mention in the talk. With that, oxygen is probably the rise of oxygen is one of the still unresolved questions when we think about the history of our planet and the really many planets that our planet has been through its history. Because even if you don't care about Earth history that much, as astrobiologists you should, but here's probably a graph that we've seen at some point somewhere. And without going into the systematics of sulfur, multiple sulfur isotopes, this is a measure of the composition of sulfur isotopes in various sedimentary materials from all ages. And what you can see, whatever the signal is, it really is scattered all over the place. up until about 2 billion years ago, and then it collapses to zero after that. And that in 2000 was used to really argue for a major change, the only change that could be assigned to this, like how do you make this huge signal and then somehow collapse it? This was attributed to the presence of ozone in the atmosphere and gave people probably the first quantitative indicator of how little oxygen or how much oxygen there must have been in the atmosphere at this point in time, meaning there was less than that at this point in time. So basically the signal is, I don't know what's going on with my arrows, but yeah. So this signal is really interpreted as the reducing atmosphere before about 2.5 billion years ago, and an oxidized atmosphere with oxygen levels that were somewhat higher than 10 to the minus 5 of the present atmospheric level, but we don't know exactly how much more. And this event, or really the time of change, is called the Great Oxidation Event at about 2.5, 2.3 billion years ago. And that momentous change, at least as seen in sulfur isotopes and certainly many other indicators of oxygenation, has been attributed to finally cyanobacteria that finally evolved and made some oxygen, enough oxygen to convert a reducing atmosphere into an oxidized atmosphere. Now, when cyanobacteria actually evolved and how long this transition was going on for is still an open question. Did it happen 5 minutes before midnight? So really right before 2.5 billion years ago, or maybe even at 3 billion years ago, and then took forever for Earth to become oxygenated. We don't quite know. And here are the organisms that could have preceded cyanobacteria. Photosynthesis is by far the most energetic, the biggest source of energy on our planet. The sun is there. It provides 10,000 times as much energy as the geothermal sources. And sure enough, some microbes evolved that are able to take up various inorganic electron donors, sunlight. They extract electrons from these electron donors, dump them onto CO2, make organic matter and oxidized compounds such as iron and manganese and various sulfur, oxidized forms of sulfur. Now, all of these processes are thought to have preceded oxygenic photosynthesis, the ability to split water. So these organisms would be evolutionarily more ancient and probably precursors to cyanobacteria. But also they were limited to environments where all of these electron donors were abundant, but not really elsewhere. Once organisms such as cyanobacteria learn to split water, take up sunlight, and dump electrons onto CO2, this really liberated microbes to colonize environments. All they need is water, of course, some minerals and nutrients, but far fewer than an iron oxidizing bacterium should use. So this is really an incredibly complex biochemical step, evolutionary step. So when did this happen? Now we can attack this problem. We can really look at this by asking, so when do first cyanobacterial fossils show up in the fossil record? So I told you the GOE, if this is our time scale, and I always look at the time scale in billions of years ago. So this is the GOE, the Great Oxidation Event. And the oldest fossils of cyanobacteria that are diagnostic, that people, serious paleontologists really say, oh, these are indeed cyanobacteria. They look exactly like some modern organisms we know. These are the fossils, and they're 2 billion years old. So they're actually younger. So that doesn't really get us far. There are various chemical signals that people have used to interpret or look for traces of oxygenic photosynthesis before this GOE event. So really deep down in the past, and really there are many opinions of what redox proxies to use, and really different levels of understanding of these different proxies, different levels of interpretations and confidence in actually measuring the chemical signals. We had some thoughts on that as well, but by and large, You can interpret these to mean that oxygenic photosynthesis maybe was present from maybe 3 billion years ago onward. Some people claim even at 3.5. So really pick and choose, but we ultimately do not understand a lot of these redox proxies well enough, and alternative processes that can create some similar signals in redox proxies. So really, the geochemistry also is unclear as to whiffs of oxygen before the actual rise of oxygen at 2.5 billion years ago, or slightly later. So another record that has been used and it's become really has improved in the last decades is the record that is stored in the genomes of modern organisms. So what we can do is we sequence a lot of modern cyanobacteria. We understand, so at the tip of this is a phylogenetic tree, at the tip of every single branch are really many modern organisms. We can grind them up, get their genomes sequenced and compare their evolutionary relationships. And really all these different branches denote different organisms that are more related to each other. And so all of these colored bars are some types of cyanobacteria. We can compare them to other bacteria. We can plot these evolutionary relationships. And now, if we have some good fossils or good ways to tie these evolutionary relationships to time, to understand when some of these clades, different clades appear, then we can produce chronograms. And from these chronograms, read when different clades evolved, and that can get us all the way down to, so if all these colored bars are cyanobacteria, that gets us all the way down to the split between non-cyanobacteria and cyanobacteria, or really the evolution of this crown group, which is all the group that contains all modern cyanobacteria. So that's the crown versus the stem, which is the line that led to the first common ancestor of all cyanobacteria. All right, so here's where we are lucky. Cyanobacteria are the only microbes with a really diagnostic fossil record, which means that there are fossils that are billions of years old that we can directly compare to modern organisms, and they really do not look like any other microbes. This is not the case for most microbes we have. So alpha proteobacteria, good luck. But cyanobacteria, there are fossils that really colonize, even found in the same types of environments that we find cyanobacteria in today. And so here are some examples. So here is this coiled fossil. All of these are silicified, so found in chert. So here is a 1.5 billion year old fossil of this coiled organism that looks just like some modern cyanobacteria. There are some endolithic cyanobacteria. They all show up at different times throughout the Proterozoic, so after the GOE, but still good enough for us to place some dates on those rates of evolutionary events. And there is this 2 billion year old Entophyssalis fossil. So these are these microbial mass that are also silicified and preserved really as diagnostic fossils with particular cellular division and multiple envelopes that look just like some organisms that are today found in Shark Bay. And if we are lucky, we have genomes of these modern organisms, modern cyanobacteria that we can sequence, or maybe they've been sequenced for us, and then we can do the magic with molecular clocks. This is actually the magic done by my collaborator, colleague Greg Fournier. And we can start looking at some rates. So here's what we did. We had to actually improve the coverage of some of these major groups of cyanobacteria that are represented by some of these really old fossils. So we had to do a lot of sequencing, a lot of enrichments to get cyanobacteria that we were really interested in, and they have good fossil record, well represented, and establish their evolutionary relationships. And that allowed us to actually place some of these fossil calibrations onto different nodes. And so when we did that, here is what molecular clock models produce. And the nice thing about all of these calibrations is that we have multiple. So by sequencing all of these modern cyanobacteria and adding more to the database, we now have the ability to play around. Maybe we don't like certain fossils, maybe we're not so sure about some other interpretations, but we can play around and pick and choose to see how different assumptions and different calibrations influence our ages. So how old are cyanobacteria really? So when we do that, and Greg also played with different models, there are different evolutionary models that go into assuming really how quickly genomes and in which way genomes change, how cyanobacteria evolve. So all of these different colors denote different models, and there are some also pick and choose points with calibrations. But any way you play this, any combination one picks gives us the date for, so these are probability functions. And so here's the age in millions of years. And the peaks of these probability functions and even the long tails are actually all before the GOE. So that tells us that cyanobacteria, the crown group cyanobacteria, predate the GOE by quite a bit, probably by 500 million years or so. So that tells us something about the evolution of our planet as well. There are these oxygenic organisms. All known cyanobacteria are capable of oxygenic photosynthesis, unless there are some weirdos that lost it for different reasons. But all of the major groups are oxygenic photosynthesizers. The last common ancestor had to have been one. And so that had to have happened at any point here, whichever model you like. And Greg also is doing a lot to constrain, to really reduce the spread of these functions. But anyway you look at it, cyanobacteria had to have been there for hundreds of millions of years before the GOE. So now the question is, so what was the Earth doing? Why was there no oxygen in the atmosphere, and why did it not accumulate quickly? And I think I'll punt that question to atmospheric modelers and people who care about volcanic activity. So just to place this in a timeline of other redox proxies, some textural proxies, so really some stromatolite textures and some chemical signals. And just to emphasize, all of this was done without any assumptions about the GOE and when it happened, and without any assumptions about geochemical proxies. So this is completely independent. Now we are comparing the molecular record to the geochemical record. So here's again, the model we thought matched best some of the constraints with horizontal gene transfer between cyanobacteria and other groups. And when we look at the crown bacteria, so this is the bacteria, the entire group, they should have evolved by, say, 3.6 billion years ago. Stem cyanobacteria, there's a large spread but certainly by about 3.2 billion years ago. Crown cyanobacteria by about 2.9 billion years ago. And here are some observations in the rock record. The oldest stromatolites, the 3 billion years old, and they are unlikely to have been made by cyanobacteria. There are some proposed cyanobacterial textures at about 3.2 billion years ago. This is the Moody's group, and this does not really jive well with this estimate for the age of cyanobacteria. So whatever that was, those may have been some precursors to cyanobacteria. There are a lot of proposed cyanobacterial textures, so just textures in stromatolites and some redox indicators of oxidative weathering around 3 billion years ago. And that could be maybe at the initial, in the beginning of this age of crown cyanobacteria. There certainly should have been a lot of aerobic metabolisms that start originating right around the same time. You're making oxygen, someone needs to figure out how to consume it. There are a lot more proposed cyanobacterial textures and oxidative weathering indicators around 2.7 billion years ago, and that certainly is consistent with this estimate for the crown, the age for crown cyanobacteria. And then what we see is that the major diversification, meaning all of these groups that have now modern representatives, really start coming to their own right around GOE, with some modern organisms such as Synechococcus and Prochlorococcus only really evolving after the second Neoproterozoic glaciation or so. So really, they are quite late. So modern planktonic microbes that are so common in modern oceans, those cyanobacteria much later. All right, so that tells us that cyanobacteria must have had a long evolutionary history. They've been around for a long time. So why do we not see them in the fossil record? Or what is it that we are looking for? And what changes? So when one thinks about the fossil record of cyanobacteria, as I said, all of it is Proterozoic, so after the GOE. And this is what it looks like. These are these diagnostic cyanobacterial fossils that are 2 billion years old. All of these, so this is silicified. What you're looking at, all of the dark material is little organic blebs that are preserved in this micro or really nano, by nanoglobules of silica. You see some remnants of former what was cell, and there are these multiple envelopes, even some communal envelopes, and these characteristic tetrads and division patterns that really tell people these are cyanobacteria. And these tetrads actually end up making clumps that make pustules, little pimples that are identical to the structures that are found today in shark base. So here are these pustular mats. I'll be talking a lot about them. This is what they look like. This is about, so these little cones are about maybe 0.5 centimeter to centimeter tall. And so that gives you the sense for the scale of these pustules as well. So we collected some of these pustules in Shark Bay and looked at the cyanobacteria because we were really interested in the modern analogs. And so here are the modern cyanobacteria that we managed to enrich and grow in the lab. You see this is a transmitted light image, cells surrounded by multiple envelopes, surrounded by all of these common envelopes. And they indeed make pustules when you grow them on a plate. So what we wanted to know, what Kelsey Moore, actually my former graduate student, wanted to know is she loved cyanobacteria, and she really wanted to understand how something that is growing, when and how it becomes a fossil. So what conditions, how much silica needs to be around, and how that happens in marine environments, because this is where our record is from. It's from marine environments, it's from very shallow peritidal environments. So here is an example. This is actually from the Belcher Formation, this 2 billion year old formation. Most of the fossils are found in little blebs and pods of chert, this amorphous silica that is enclosed within dolomite and within these entire structures that resemble some modern structures made by pustule forming cyanobacteria. I think these are from somewhere in the Gulf. I can't remember exactly. So these are modern, these are ancient, but really the same size, shape, and the environment where they occur. So really these peritidal, evaporative, really exposed environments. And usually these old ones are in dolomite that encloses chert. So Kelsey was really wondering about all of these associations. Why is it, what makes dolomite? Why does dolomite end up assuming the shape of really the microbial mats and making these stromatolites? And why only in some places we see this chert? Here's another example that I'll be talking about later that is 1.6 billion year old Balbirini dolomite. So another example of very exposed environment, dolomite that encloses chert pods, and the dolomite and chert are really intimately interwoven to the extent that the dolomite will preserve some of the microbial textures, so some of these lamina in the stromatolite, and little pods of chert will preserve fossils. So they preserve textures, but they also preserve body fossils. You don't find fossils themselves in the dolomite. So if you look at these have been described in the 70s. These little blebs here are actually full of fossils. And they are chert, and this is what these fossils look like. So again, these characteristic multi-layered envelopes, the goop that these organisms secrete, and enclosed in silica, and also many, many cells within these colonies. So if you think about modern environments where silica precipitates, we really go to hot springs. Here is an example. This is actually my first graduate student, Minsab Sim. We did a lot of work in Yellowstone, and here he's looking at a hot spring, and all of this white stuff is silica, amorphous silica that precipitates and actually encloses and fossilizes some of the modern cyanobacteria, all of this orange stuff here in Yellowstone. And that's a hot spring. Hot fluids dissolve rock and they bring silica to the surface, and then the fluids cool down, and as they flow from the source to these outer regions, and cold solutions don't contain so much silica, silica precipitates. But how is that really a good analog for a marine environment? And so what is different about the Proterozoic marine environments in terms of silica? They had to have contained more silica, no diatoms, no silica precipitating organisms, but how much more? And why also do we find all of these carbonate minerals together with silica in these marine environments? They typically occur together in a lot of these fossiliferous deposits. So what Kelsey decided to test with experiments she really was probing all of these questions. She was wondering how cyanobacteria become silicified. And so really the question was, can silicification of cyanobacteria by amorphous silica occur at roughly surface temperatures under just normal pressures in seawater? So not a solution that flows out of a hot spring, not fairly freshwater solution. And in a solution that is undersaturated with respect to silica. In Yellowstone, silica coats anything. You have a leaf, it'll just precipitate and coat it. And because it's so saturated. But that may not have been the case in seawater in the Proterozoic, because all of this preservation is patchy. There are only pods of silica here and there. So it's more of a special condition rather than a common condition that makes silica precipitate. Then another question was, well, if these fossils are so beautiful, so here we are looking at 2 billion-year-old fossils, multiple envelopes are again seen. So what is it about cyanobacteria? And usually it's cyanobacteria that are really recognizable and preserved. So what is it about them? Do they play some potentially active role in silicification? So is it something about their metabolism? Or is it just that they are surfaces and they just get coated? Which really, this is similar to asking which biological factors or chemical factors. So what solution compositions influence silicification? And then the question of ecology. Does the presence of other organisms, I was talking with Norm in the beginning of the seminar, and in nature, there are not just cyanobacteria. Microbial mats are vibrant communities of many, many microbes. So what other microbes in these communities actually matter for the preservation of these biosignatures, fossils, textures, and so on? Do they matter? I mean, probably not a question to ask a microbial ecologist, but I certainly ask that all the time, because we need to know what it is that we are taking out of the system, or what we can take comfortably, or should we just study cyanobacteria? All right, so Kelsey developed a system in which to ask all these questions. So she had some cultures with seawater. She enriched these cyanobacteria from Shark Bay. This is, again, what they look like. They have these EPS envelopes. She figured out a way to grow them so they have more of this exopolymeric substances, material. We just call them that because there's a lot of stuff, mostly sugars. And she also developed a floater system with the help of some postdocs in the lab. And the system is important. So what she does is she had microfluidic chips, and she puts pieces of these microbial mats, mostly cyanobacteria with some other organisms, on the surface and flows this seawater. So now it's seawater, it's not fresh water. She adds more silica than one would find in modern day seawater, but not enough silica to have abiotic precipitation. So no precipitation of anything occurs unless there are bacteria. And she has a pump, peristaltic pump, and she just keeps flushing these organisms which are growing with a solution, and then she can sample them at particular intervals and look at what happens. And so importantly, again, there is no, unlike in modern hot springs, there is no abiotic silica precipitation. We are trying to really emulate a marine system. And so what she found, so here is a time sequence. So she starts with these cyanobacteria. These are SEM images, scanning electron micrographs. And all of these organisms, so there's no mineral here. The surfaces are wrinkled because all of these polymers get slightly dried out during the preparation. And there's no, so if you look at the composition here, there's no silica really to speak of, mostly just carbon, oxygen. There's quite a bit of sulfur here, which we'll come back to later. In 15 days, she sees that the silica peak increases, and she does see slightly, less wrinkling of the surfaces, meaning they're getting impregnated by something. And then after about a month, she sees the surfaces that are coated with tiny granules, large silica peak. There's quite a bit of magnesium too, and tiny granules that are actually this amorphous silica. So silica is amorphous. You can't really test it for its presence by XRD, but what we did was use FTIR. And so here is a comparison of a silicified biofilm, so silicified cyanobacteria and unsilicified. So this is day 0, day 1 versus day 30. And what we see in day 30, so the silicified biofilm, that has a strong silica signal, we do see some characteristic peaks that tell us that amorphous silica is really present. So these are these 2 peaks labeled, so really confirming the amorphous nature. This is not some weird clay mineral that contains silica, it's really amorphous silica. And so having this experimental system allowed Kelsey to actually probe what concentrations of silica worked best, really allowing her to probe into this potentially ancient seawater, what was needed to make fossils. So here is an experiment where she used 70 parts per million. This is maybe comparable to some places in modern oceans. 70 parts per million certainly higher, and 100 parts per million higher. And so you can see this roughness. You're looking for these granules. The best preservation, the most impregnation with these silica granules happens at 100 ppm. 70 is still very good, and at 17 really you see a lot of wrinkling and not much silica. So really 70 ppm silica or more seems to be the best, which gives us a constraint for the concentrations of silica that had to have been present at least very locally to have fossilization happen, to preserve those beautiful fossils in seawater. And all of these concentrations are below the saturation. So again, no silica precipitates unless we have cyanobacteria. So what is it about cyanobacteria? Is it that they photosynthesize? Is it that they are producing these envelopes? And so Kelsey tested that by just incubating, taking cyanobacterial biofilms and putting them either in the light where they grow, or just covering the whole system, wrapping it in aluminum foil. So they were in the dark, and looking at what happens after 30 days. And in the light, they photosynthesize, they even grow, they keep producing this fresh exopolymeric material. And you see this huge silica peak, and again, these rough surfaces, whereas in the dark, they decay, degrade, and there is not much silica that is bound at the surfaces. So activity does seem to promote better silicification. So for this process, we do need to have active surface organisms, active photosynthesizers. And by looking at more of these organisms and samples, so here is really a collapsed section or cross section through one of these colonies. So these were remnants of 2 cells. This stuff here is organic, just a collapsed cell. And all of these layers that are encasing these cells are those multi-layered envelopes. So each of these cells had its own, and then there was a common one. And when Kelsey mapped where silicon was, it really always occurs not in the cell in the interior, but primarily on the outside. So it seems like that material, it seemed to us that this material was really good at binding silicon. And to test that further, we just extracted it. And so when Kelsey extracted this material from cyanobacterial cultures and just incubated the polymer, it's polymeric material, she added silica 70 ppm or 90 ppm, I don't recall exactly, and compared that to the controls where she added the same amount of silica but no extracted polymers. And she noticed that silicification occurred only in the presence of polymers, but it was just in the presence of the polymer. So polymer itself can promote silicification. So here's this peak of amorphous silica and this silicified in really a jar that contained extracted polymers. And as I mentioned already, in all of these cases, we would see an increase in the silicon peak and a magnesium peak. That seemed interesting. It seemed to us like some amorphous magnesium silica phase kept precipitating. It's amorphous silica, but it has a lot of magnesium. And Kelsey tested that further. What is it about exopolymers around the cell? So it's really those envelopes. All of that goop that they secrete seems to really actually be really good for fossil production. And she noticed, and she really measured a lot of, she measured the responses of that material to magnesium. It binds magnesium itself. And so she came up with a model of a cell that produces this extracellular polymer, the envelope that contains a lot of actually sulfate groups, also some carboxylic groups, binds magnesium cations, and that magnesium forms a bridge that then binds the negatively charged silica that's in the solution, forming these basically bridges. So this is how cyanobacteria seem to be able to silicify in undersaturated seawater and really become fossils when they are active. To test that further, the prediction of this model, so in this experimental system with these pustular mats, Kelsey just saw a lot of magnesium. That was usually the peak that increased. And the prediction of that model is that, well, if we have some really beautiful fossils, meaning well-preserved, the silica around these fossils, around the organic matter that is preserved, should be enriched in magnesium as well. And so we actually communicated with, I think it's not the Geological Survey in Australia, it's a museum in Canberra. And they have thin sections. So these are museum quality thin sections of this Proterozoic shirt. It's in the Balbirini dolomite. It's about 1.6 billion years old, where some of the beautiful pustular cyanobacteria were described in the 70s. We got some of these samples, and Kelsey, now postdoc in Ken Williford's lab, mapped out the silicon and magnesium and also calcium, because there's a lot of dolomite here. And so here is a region that she mapped. The dark stuff is organic matter. So this is now a backscattered electron image. So dark is organic. All of this maybe slightly darker gray is silicon, and the surrounding areas are dolomite. So really we are talking about very small areas where silica and dolomite are present. And so here is the map of carbon. Here's the map of silicon, and here are the ratios of calcium and silicon. Calcium will occur in dolomite and really nowhere else. But magnesium, interestingly, is enriched in places where silica is present and calcium is not. So it seems to confirm the model. And so these are these pustular mats. And so it does seem to indicate the presence of similar mechanisms that were responsible for the silicification of these similar organisms with similar lifestyles for a long time. Now, Kelsey extended that to analysis of other Proterozoic fossils. For example, some fossiliferous sections from the Duck Creek Formation, again, similar mapping. All of this now is silicon with carbon areas that are mapped, magnesium, aluminum, calcium. And she saw that not only magnesium, so these contain more filamentous fossils, and she saw the enrichments in not just magnesium, but also areas with a little bit of aluminum and also calcium, suggesting that maybe some organisms also tend to bind other cations that promote silicification. So this is the suggested model, and now we need to test a lot more organisms. And we started by actually looking at mats or structures that grow side by side, really a centimeter away from the pustular mats in Shark Bay. We brought back some of these tufted structures. So they're about maybe a centimeter tall, and they look like little Christmas trees, and we enriched some cyanobacteria from them. This is what they look like in the lab. They really, these are ropes made by many, many cyanobacterial filaments, so not pustular mats anymore. And we just wondered, Kelsey was guiding a thesis by an undergraduate, Kyle Morgenstern, who did all of these experiments. And he found out that these bacteria incubated under the same silicifying conditions, silicify really, really well. So here is one of these, imagine taking one of these ropes and taking it under the SEM. And so each of these ropes that is visible is made of many, many, many cyanobacterial filaments. This is what they look like when they're aligned. And all of these surfaces on the outside, so this is the outside, here is the inside of a rope. All of these surfaces on the outside are rough and meaning silicified, huge silica peaks, whereas the interiors themselves are not. So again, the surfaces of these cyanobacteria seem to silicify really well. And interestingly, unlike the pustular mats, there is an enrichment. So there's silicon peak, but there's also quite a large calcium peak and maybe some small magnesium peak. So it seems like it matters what kinds of bacteria are present, what kinds of cyanobacteria are present. They may have different surfaces that tend to bind different cations. So that tells us about potential also silicification or taphonomic biases. Not all cyanobacteria may be able to silicify well. We may not get fossils of all types of cyanobacteria that are even present. And to test that, Kelsey decided to compare the Shark Bay pustular mats with some organisms that make exactly the same structures and have the same kind of the similar envelopes and really same shapes and sizes of cells, but come from some freshwater environments. And she grew the marine cyanobacteria, the Shark Bay ones, in marine medium. And you look at all these rough surfaces again, all of these silicifying conditions, undersaturated. And she grew the same freshwater cyanobacteria in the marine medium. It does just fine. You can toss it in salt water, it'll be just fine. And very little silicification. So some of this roughness, but very little. And she also grew the same cyanobacterium in its freshwater medium with the same concentration of silica. So freshwater medium, not much magnesium, no sodium, no chloride, or very little, no silicification, the same concentrations of silica added. So it seems like different cyanobacteria really do have different silicification potentials depending on their surfaces, because at least one of the differences in this exopolymeric material that we noticed when we compared, so this is the brown organism EPS, exopolymeric substances. The brown one is the freshwater one, and this blue one is the Shark Bay one, marine hypersaline, in fact. And we see at least one major change between these substances that are excreted on the outside. The marine organisms contain a lot of sulfate functional groups. All that sulfur we were seeing in the electron dispersive, X-ray dispersive spectra, it matches, actually, it seems to correspond to the sulfate that is present on the outside of the cells in these functional groups that modify sugars. And we are now wondering whether these sulfate modified sugars have particularly important role in silicification. And none of these are actually present, these very indicative groups, in the freshwater organisms. So at least that is one of the differences among their EPS. And the presence of these envelopes and all of that material that is so important in making fossils depends on conditions. So here is why we go to, I mentioned all these peritidal exposed environments where we find the best fossils. Not only would these be the environments where you dry out things the most and you concentrate silicon the most, but these environments are actually also places where only some cyanobacteria can grow. And it seems like only those cyanobacteria that are able to excrete very copious exopolymeric substances. So here's what happens when you just grow our cyanobacteria from the pustular mats in liquid culture, no UV. You see cells, there is a little bit, this halo is a little bit of EPS, and that's it. Not much. Here's what they look like when you grow them on a plate in the presence of UV. This EPS, this compound, the biological mechanism by which they silicify, actually is induced in a stress response. So stressed organisms silicify better, it seems like, and produce compounds that, including the sulfated polysaccharides that make them particularly resistant to those environments. And sulfated polysaccharides typically are associated, you'll find them in all sorts of algae and food additives like carrageenan and alvin, so all of these good seaweeds. But cyanobacteria, I think we need to start paying attention to what all of these microbes are doing in extreme environments and how they are, what their chemical properties tell us about the stresses they experience, because sulfated polysaccharides seem to be secreted, particularly in response to water stress, desiccation. And so to understand more about these polysaccharides, so here now we are switching from silicification to precipitation of carbonate, or really how these mats survive. So here is a mat that we collected in Shark Bay. And in Shark Bay, this is what they look like. There are a lot of cyanobacteria on top, so all of these dark areas, cyanobacteria, all of this shiny material is bound particles or precipitated particles of calcium carbonate and microbial goop. These are very squishy. So Emily Skoog, graduate student, sectioned them. And here's just a thin section. So here's the scale. It's really the same mat. So this is the section they analyzed. So Emily and Kelsey mapped this by backscattered SEM. So you see dark areas of cyanobacteria, the particularly bright areas are where calcium is, and some of it precipitated. And this slightly, I hope you can see this, but these bright areas, actually the sulfur is the sulfur in EPS, seems to track really areas where cyanobacteria are. So this is this fresh exopolymeric material, whereas the degraded material does not. So these areas that don't contain so many cyanobacteria, you don't see so much sulfur and you see much more calcium. So it seems like this is something to explore now. It seems like microbial processes that degrade, it's almost like an outside gut. There are a lot of organisms that eat the matrix that cyanobacteria produce. And that eating of that matrix helps the rest of the mat actually precipitate calcium carbonate. So we are now approaching this by a lot of sequencing of these communities and looking for enzymes that are particularly good at eating the matrix. So who is eating all of this goop, and looking at who is producing it. And cyanobacteria, indeed, if you look for genes, I won't go into details, cyanobacteria are indeed the major producers and really the only producers of sulfated polysaccharides in the system, all of this sulfur that is mappable onto the actual mat. And it seems like this is the work done by another graduate student, Elise Katz. So what she found was that there are all these other organisms in the system, and it seems like when you look for enzymes that are excreted out of the cell and degrading polysaccharides, all of these polymers, Bacteroidetes are the microbial group that seems to be important in, they're the first degraders of all of these extracellular polysaccharides. So if you think about the fossilization potential of microbial mats and how that can all work together, or how it worked together in the past. So this is the Balbirini dolomite thin section that you've seen before. How do we get silica? When do we get silica? What conditions are necessary to get silica? And then carbonate. It seems like to get silica, so areas like these that actually exhibit fossil preservation, preserve actual fossils of cyanobacteria, you always just see these cyanobacteria there. And so to get the preservation of these types of mats, we need photosynthesis that's active. It increases the pH and stimulates the precipitation of silica. We need fresh cyanobacterial EPS, so fresh surfaces, growing active cyanobacteria. So silicification does seem to happen more on the surface of the mat. Whereas if you're thinking about where dolomite or calcium carbonate precipitates, it seems like this is the calcification process that happens deeper in the mat away from these active zones where silicification can take place. And that should be the function of pH and EPS degradation. And this is why it's really important to look at organisms other than cyanobacteria in mats. They are needed to preserve the record of communities deeper down in the mat. And this would happen in the interior. This process does not preserve body fossils, really. Now, if you're thinking about the evolution of the environment, so we've been able to say something about a silica, put at least an upper level for the concentrations. It seems like the seawater in marine environments in the Proterozoic was not super saturated with respect to silica. And there was at most, given the patchy nature of preservation, really only in the most evaporated places, it was higher than 60, 70 ppm, which means mostly it was much lower than that. But then if we think about these physiological responses, the organisms that are best able to colonize exposed environments, secrete these polymers, secrete sulfated polysaccharides, they need sulfate to do that. And it seems like the marine sulfate, really, this is from some recent work from Clara Blattler, who did some modeling to constrain the concentrations just using these samples of cores. I think these are 2 billion-year-old cores. And she was looking at anhydrite, so sulfate-containing minerals, and she constrained the sulfate concentrations to be around 10 millimolar at 2 billion years ago. So it seems like conditions at that time become just right for us to have a beautifully preserved fossil record of cyanobacteria. So perhaps they were present before, but not either not colonizing the right places or really not having the ability to adapt to some of these most exposed places before that time. So all of this, I'll just send, this is my one requisite Mars slide, and I'll just say that all of these mechanisms that I mentioned and some additional ones are important now for the search for biosignatures or any just preserved organic matter in Jezero crater, because all of these phases are present. We have carbonates in, this is the landing site, Jezero crater of the Perseverance rover. The carbonates are present in the rim of what would have been the lake. There are some clay minerals. There are actually patches of silica, even amorphous silica detected from the orbit. So we may encounter some places that we may want to sample for even microbial fossils. We can talk about it more. So I'll just end with conclusions here. I'll stop sharing and open it to questions. I think I summarized it.
All right, wonderful. Everybody throw up your clapping hands emojis to show your appreciation of that excellent talk. We have a couple of minutes for questions. As always, there are two ways to do that. You can raise your hand using the reactions tab, or you can type your question into the chat and I'll read it aloud for Tanja. So either one of those methods works.
I can stop sharing this, but yeah,
okay.
I can see.
I guess while other people organize their thoughts, I'll ask a question of my own. So you showed some really cool images of this communal nature of cyanobacteria and how that's a very unique indicator of them. I was wondering if that has any connection whatsoever to their unique metabolism. Why do cyanobacteria seem to organize themselves in that interesting way?
These are so there is a bias when I talk about fossils. There's certainly a bias towards fossils from benthic environments. This is where you deposit minerals. That is really where the record gets made. So then we almost by default have to ask what were the organisms that colonized the surfaces? What were they like? There are some organisms deposited maybe deeper in shales, but really by and large, the record is of benthic organisms. And to colonize a surface, you do need to attach yourself to the surface, and you do need to, and if the conditions are right, something again that Norm and I were talking about in the beginning, there's no bound. If you can survive the UV, if you can survive, if you get enough water to live, organisms multiply. The conditions are right. They need light, and they are not buried by too frequent sediments. They just can spread, and so you can get extensive surfaces and really many, and they are microbes, so they really need to. There need to be many of them to see them.
All right. Excellent. So now we have 2 raised hands. We'll go with Norm and then Anitra, and I think that's probably all the time we'll have. So these will be our 2 questions from the audience. So Norm, why don't you go first?
I'll be quick. Your 10 millimolar sulfate's about a third of the modern level. Yep. Would that be an open ocean level, or has it already been concentrated from the open ocean level in your environments?
So this is, I have to go back to the paper. So the deposits are from a hypersaline evaporative environment. These are evaporites that they sampled for sure. But I think they did some neat isotopic balance modeling to really constrain the calcium to sulfate ratio and so on. And I think that's how they arrived at these concentrations, the upper limit for the concentrations in the open ocean, if I remember correctly.
Okay.
Yeah. But these are what they sampled are evaporites for sure.
Okay, but it is just anomaly and extrapolation back to those. That's nice.
Yeah. Yeah, I need to go back. I read a paper and it was really, I thought they really did a neat job and it seemed convincing to me, so.
I got 10% of PA modern level for very convoluted reasoning way back when, but if somebody could get, I presume there would be a better way to do it than I did.
Yeah, they have also now.
They have the rocks.
Samples and yeah, they could do quite a bit to constrain different.
I'm just considering the mass balance at the ridge axis. So.
Yeah, so
okay, it's good getting it.
Yeah, it seems high. It seems high though. You're right. It's like
it seems high to me, but it should be. I was getting the point where you start to form anhydrite in the downwelling at the ridge axis. That was the number I gave, and but getting it multiple ways is good, and having it.
Yep.
Norm, you just muted yourself.
I just muted myself. It means my question is over. Sorry.
All right. Anitra, take us home.
Yeah. In the modern ocean, the major silicifiers are diatoms, and it's known that they use organic molecules also to precipitate silica, and they're like polyamines and proteins, and oftentimes they're sulfated as well. And I wondered if you had thought about parallels or thought about, evolution of this process and how it might have gone from cyanobacteria to other things and the role of these template molecules.
So one major difference between cyanobacteria and diatoms is that the cyanobacteria, from everything we've seen, do not tend to concentrate silica. Because diatoms are like little sponges. They just grab every molecule they can find. And this is why the concentrations are kept low. But I think the silicification, and I think there are certainly different types of, polyamines, those silafins, all of that beautiful stuff. I love that paper. And there are probably different types of organic surface. Something we are really curious about, to what extent is this sulfated polysaccharides? To what extent is it, there is other stuff in EPS. There's some proteinaceous stuff. but also I think silicification is a process. When you look at how sponges do it, they do it differently. And so I think there are multiple independent origins of the process based on everything we know.
Yeah, it seems like the sulfate plays a role in maybe in all of them, I wonder.
If there are some, yeah, I don't know how much, I haven't seen much about sponges and sulfate. I know people have talked about chitin and some composite materials that help silicify. Actually, you even have calcium carbonate and then silica around all of that. But I think there are multiple independent origins of the process. And I don't think cyanobacteria care. I don't think they care to silicify at all. They just happen to be surrounded by enough silica, and that's what happens. Although, okay, this is a tangent, but if you silicify, I mean, silica, given where these organisms live, if you have enough silica around, silicon is what we add. We don't grow them. If we want to expose them to UV, we don't grow them. We don't cover them with glass. It's a known, amorphous silica is a known absorber of UV. And so it could help them actually, because they live in these very exposed environments.
Right.
So something we are hoping to test soon. We want to do some experiments. Before COVID hit, we wanted to do that.
Great. Thanks. Nice talk, Tanja.
Thank you. Good to see you.
All right. Unfortunately, that's all the time we have. So let's thank Tanja once again. That was a wonderful talk. And we'll see you all back here next week for Sonia Tiku-Schanz's talk from Stanford University. Take care, everybody.
Thanks for inviting me. Good to see you all. Bye.